So the next speaker is Margit Ross. Margit has a master's from Drake University. She has a scientific background spanning 37 years at Pioneer, International, DuPont, and Corteva Agriscience. And the predominant areas of research focus have been involved in elite maize transformation technology and also maize site-specific recombination approaches through design. The title of Margit's presentation is NGT Products, Methods and Matters of Detection. Thank you very much. We look forward to your talk.
Thank you for the opportunity to speak today. I'm happy to be here. And today, perhaps my talk is a little bit different because I'm going to talk about an actual edited product for waxy maize like Ray introduced. Waxy maize was produced by Corteva in 2015. It went through a MI-regulated APHIS check at USDA. We were confirmed that it was not a regulated trait. This was also, consultations were done in Argentina, Chile, and Brazil, also with confirmations of not being regulated. So some pre-commercial trials were used. And today I'd like to talk about both the development of waxy maize, but then in relation to both GM detection methods and the possibility of NGT detection methods. So before I start that, I want to talk a little bit about Corteva. Our company is dedicated solely to agriculture. We enrich lives for both the farmer and the consumer through 3 different areas, through seed, crop protection, and digital technologies. So we have a global presence in 140 countries with research centers and production facilities within those countries. We have about 21,000 employees. And our products, from a seed perspective, which is where I come from, are both conventional and GMO. So the seeds facility is located in Iowa, which is in the north central part of the United States. Iowa is a very agricultural state with 85% of the land dedicated to farming. So you see lots and lots of corn and soybeans. So for today, first of all, I'd like to talk about the waxy maize from the CRISPR, but first give some background because waxy maize is also a conventional product that is produced. So waxy maize as a seed has an opaque endosperm, so it looks kind of like a candle wax. And that's due to the high levels of amylopectin within the seed as compared to a normal maize kernel, which is more vitreous in appearance and translucent. And it has a more balanced approach where the amylose production is at 25% and then amylopectin is at 75%. So waxy maize as a conventional product has been cultivated in the US since the 1940s. It's a specialty crop. It's grown in an identity preserved system or a closed loop system, and it's managed by the grower, so it's usually the grower directly to the processor. It represents about 0.5% of commercial corn that's grown in the US annually. So if you think about that, in the US, there's 90 million acres dedicated for corn production, so that's about 450,000 acres total. And in Iowa, an average farm has 400 to 600 acres, so about 1,000 farmers would produce waxy corn. There's a limited amount of waxy corn that goes for export or for feed and livestock. Waxy corn is also in the Asian countries a food product. So one of the major products that's produced from waxy corn is cornstarch. And it's an essential function in the food industry as a thickener agent. So when you have the high amylopectin, it creates thickening or gelling. It's also used in the paper-making industry and then the textile industry as an adhesive. So the waxy maize is caused by a mutation. And if you look in the maize GDB database, it's due to a loss of function of the waxy1 allele, but there's greater than 200 waxy1 entries as mutations representing deletions, insertions, or translocations. The deletions are variable in size, And this is the whole list, but there's many more that aren't characterized. So for CRISPR waxy, how did we make it? So the way that that was done was through microprojectile bombardment of immature embryos. And we used 6 plasmids all mixed together. One had Cas9, 2 of them each had the guide RNA, one had the MPT2 selectable marker, and then there was 2 developmental genes that, when expressed transiently, induce somatic embryo formation. So while these were in vitro, there's typically you would see a callus phase for plant or for transgenic recovery, but in this case, somatic embryos formed and germinated directly into plants. So from the BC0, or what you would call on a transgenic a T0, those plants went through molecular characterization, and we used next generation sequencing for that. To look for the absence of the inserted plasmids. And at Corteva, we use southern bisequencing, which is just a different technique to use sequence capture technology with deep sequencing. We use capture probes that covered the entire sequence of all the plasmids that were used during transformation, and then looked for novel junctions of the plasmid within each plant. And in the ones, we had none that were identified. The positive controls that we were used for this analysis were the plasmid spiked into the wild-type maize, and then the negative controls were just wild-type maize. So in this, we were able to edit 11 different elite germplasm lines. We also looked for off-target mutations using both PCR amplification and next-generation sequencing. The guide RNA that we used were conserved guide RNAs for the germplasm that were tested. And for the off-targets, we had internal software that we used to look at reference sequence for predicted off-target sites, but then we also used the Cas Off-Finder public software to look for off-target sites. And then from those, amplicons were made into probes, and that's how the next generation sequencing was done. So you can see on the slide that the only thing that we could pick up were endogenous promoters. In this case, that was a ubiquitin promoter. Okay, so because we were carrying out this waxy maize quite a ways, we were interested in looking at how can you detect this in a food product. And the normal food product from waxy maize is cornstarch, so it's an easy thing to test. So we had conventional maize from within the Pioneer brand that has a 30 base pair deletion. That's caused by a mutation, and it's one of the more common mutations that's used both here and in China. And then we had our 4KB deletion that was created through the gene edit. So both of those caused a gene disruption. Gene disruption causes that high amylopectin production, and then the end result was waxy maize. So they both did the same basic, produce the same phenotype based on the same premise. So the first hurdle that we had to look at was, could we get DNA from cornstarch? So cornstarch is not the easiest thing to work with. We received 3 different cornstarches from a commercial manufacturer, and we didn't know what mutation would be present in that cornstarch. We also had cornstarch made from our waxy CRISPR maize, and then I bought cornstarch at the grocery store, so what you would use at home. So one of the problems that we had is that when you add a buffer to cornstarch, it turns into a nice firm jello or gel. And it's really difficult to ungel it. You can't heat it. It's just like if you cook with it, it's pretty firm. So one of the things that we did was we had to use a digestion with alpha amylase just to release the DNA. We still had some gelling, but we could get DNA off of that if we used a DNA extraction procedure that was specialized for food. One of the samples, though, did not give us any DNA, and that one was a highly processed cornstarch, of which there was chemicals added during the processing part that most likely removed all the DNA or degraded it. So could we distinguish waxy mutations? Once we had DNA that we recovered, we could prove that we knew we had enough DNA using the endogenous reference gene HMGA, but we were interested to know, could we pick these up by PCR? So we looked at the commercial corn starches and then the CRISPR waxy starch. And the first one that we did was with an intact waxy gene. So intact means that we went across that 30 base pair region just to see if we could see an intact gene. And in that, we could detect it in the untreated cornstarch and the grocery store cornstarch, but not in the CRISPR, which you would expect, and not in the high amylose cornstarch, which you wouldn't expect. And then with a 30 base pair deletion assay, we couldn't detect that in any of them. And then with the CRISPR assay, we only detected it in the CRISPR. So in some cases, this was pretty straightforward, but in order to have cornstarch at the high amylose, you wouldn't expect you would have a mutation in the waxy gene because it's caused by a different mutation. So what was that? And that's a question because we only tested 3 assays, but yet we had 1 unknown. All right, so I'm going to switch gears for just a minute, and then we'll come back to CRISPR waxy. But in GM detection methods, there's always, at least in my experience, challenges and hurdles that happen. And so I'd like to share a couple examples because in a GM method, you have a unique sequence to work from. So in an edit, you might not have a unique sequence. So can those two overlap? All right, so a couple of areas. PCR platform selection. If you develop on one platform or multiple platforms for robustness, but that's not what the validation process uses, can that cause problems? If the base master mix is optimized against the platforms that you use during the development process, but they're not the same as the platforms that are used or not really designed for the platforms that are used at the validation process, this can be a problem. Nucleic acid modifications, Ray spoke about those, and it's true that they're needed to increase sensitivity at some time, but the thing that you have to remember, and from a trait developer standpoint, is that some geographies do not accept nucleic acid modifications. So while you think that they increase sensitivity and they're beneficial, if the country that we submit to lots of different countries, not just the EU, so from a practical standpoint, we want one detection method. We don't want to have multiple detection methods going to different geographies. So we can't use nucleic acid modifications. It poses a limit if the regulators in other geographies say no. The event sequence. Some events are friendly and some events are not friendly to work with. And that makes, if the sequence region is difficult, at 1.1% mass-to-mass quantification, it makes it very hard. And then the zygosity ratio. So for corn, you have a male and a female, and the hybrid seed each has a different zygosity ratio. And that ratio is based on breeding practices and then quantification by copy number analysis. The molecular conformation needs to align to the breeding. So we're going to look at if you have any of these parameters, they're all interrelated to the detection method. A single parameter or a combination of these parameters that go awry can negatively affect the trueness and precision. So if they can't be rectified, the detection method is at risk. So in summary, with a GM method, the validation process is not that straightforward. So let's look at one example. And this is a real example. I've had this happen with 2 events between the GM zygosity ratio and digital PCR. So as an applicant, we have the GM parent. We know what the parent is. We know its GM contribution. We have very careful breeding practices at Corteva, and we test at every generation. So we use that reported contribution in our copy-to-copy calculations. So then when it goes to the validation process, it's understood that the ratio is verified by digital PCR, that digital PCR method is not required by the applicant. So the quantitative method that is provided is imposed into that digital method. That ratio may be different than what you have reported or used during the development process. And so the ratio is used then to determine the work. So if the parent contribution that was stated does not match the calculated ratio by digital PCR, the risk for the actual quantification at 0.1% is very difficult. So now if you think of those complexities and then you couple that with an edited product like CRISPR, maize, what can happen? So what's its potential to be able to quantify at 0.1% mass to mass? It has to have detectability, which is the ability to state something is present or absent, and it's possible by qualitative PCR. I mean, in this case, we were looking at a 4 kb deletion. It was straightforward. However, you have to remember that each elite inbred had SNPs in it, so you had to be very careful to know where to make that actual amplicon because of the variance across the genomes. It was applicable to known edits only. So some of the challenges are the basic challenges that you would experience with a GM detection method, no different. Identifiability is the ability to distinguish something from another. So is it possible that it's applicable to known edits or known sequence? So we had a 4 kb deletion as an edit. Is there a mutation out there that's a 4 kb deletion? It could be, I don't know. Could we identify the difference? I don't know. I mean, we knew that we gave maize waxy from CRISPR to a starch manufacturer, and that's what we received back. But that's because we knew what we were working with. So some of the challenges, like I said, we don't know if we could discern it from the mutation. The germplasm variances may have other SNPs present that may confound the data. Common genetic elements, like Ray said, they just don't exist. And there's no high throughput method. So really, you should have the ability to look for all mutations so you can discern across them, and you can only do that through sequencing. But there's so many of them with this example that you really need a high throughput system to be able to do that. So from a practicability standpoint, how feasible is a quantification detection method for an NGT product? So specialized PCR, I already told you my story about you can't use nucleic acid modifications. Not that you can't use them, but you can't submit to other geographies for them. And then the ease of transferability and repeatability. Some of the nucleic acid modifications are actually harder to work with. Sensitivity, I think we heard this from Ray too, small changes in bulk grain may be unpredictable. You know, for what I had with CRISPR waxy, that might not be the case, but for other single nucleotide changes, you might not find it. And then discernment is probably one of the bigger ones. How do we distinguish an edit from an other natural mutation in current products? The sequence variation across the germplasm, and then competition with other species of DNA in the food. Think of if cornstarch was in another product, another food product, could we find it? I don't know. And then what do you use for certified reference material? And then the reference sequence. So if not for corn, but for other species that have subgenomes, how would editing, if you had multiple subgenomes, would you edit each gene across those genomes, or what if you only edited one? Could you still detect it and discern? The sequence region, like I said, even for a GM trait, may be difficult. It happens. I've never had an experience at Corteva where a criteria was used that for moving an event into a regulatory approval process because it was easy to deal with on a detection method. That just isn't a consideration. You have to have knowledge of the sequence change. And then the variability of the DNA replication over time. As breeding processes change, or the plant matures through the breeding cycle, you always are going to have variation. So if you take all those GM complexities and then you couple it with the potential of edits, complexities that happen, you have even more parameters that may be problematic for detection at a quantified level. And even if you detected and were able to quantify it, it still may be impossible to identify or distinguish the exact source of detection for some of the gene edits. Okay, so for quantified detection of NGT products like CRISPR waxy, it's important to give consumers a choice in their food products. So think of cornstarch, and there is non-GMO cornstarch, but cornstarch is from a mutation. So now what happens if we would ever decide to commercialize waxy cornstarch? Is it fall into which category? A GMO? Or is it still a mutation that is like cornstarch? Something to think about. Yep, I'm there. So is it a could it be a fit for purpose detection method? It may require specialized PCR or special handling. It may be impossible to confidently assert. There's potential for this product that I talked about, but you cannot expect it for all, and uncertainty exists. So in conclusion, certain NGT products may be able to have robust validated detection methods, but not all. You have to make a fit-for-purpose detection method, you need to be aware of the gene edits and have unique sequence recognition. And you have to be able to carefully test things. And to date, there's no high-throughput genome screening method to use, nor common markers. And so this yields to overall uncertainty, like we've talked about in the last 2 days, for specificity and unique sequence recognition. So with that, I thank you, and thanks again for letting me speak.